Thank you very much for the kind introduction. Welcome also from my side to my talk, in which you will learn how to apply targeted metabolomics to study neurodegenerative diseases, but also which role the gut microbiome plays here. So what can you expect to hear in the next approximately 30 minutes? In the first part, I will introduce you to the gut microbiome and its role in different diseases, as well as the use of metabolomics for microbiome research. And in the second part, which will be our focus today, we will discuss the link between neurodegenerative diseases and the gut microbiome and the importance of the metabolism here. In the end, we will summarize today's learning. So stay tuned and find out how you can apply targeted metabolomics in neurodegenerative diseases. So let's start with a brief introduction to the human gut microbiome and how you can study it with metabolomics. First of all, why does metabolomics matter? We will see here why the metabolome is that part of the omics cascade known to be closest to the phenotype. The metabolome of an individual is unique and is influenced by a multitude of endogenous and exogenous factors. Endogenous factors comprise the genome, which remains relatively stable across lifetime, and also the transcriptome and the proteome. But also your residential microbiome can be seen as an endogenous factor that highly influences your metabolism. Exogenous factors that impact your metabolome, but also other levels of the omics cascade, especially your transcriptome and the proteome, and also your gut microbiome, are collectively termed the exposome. This includes, for instance, the exposition to external pathogens, the environment we live in, your lifestyle, the drugs you take, and of course your diet. The metabolome, in fact, integrates the genomic information and all the other influencing factors to provide a functional systemic readout of the present phenotype. And so, metabolomics is a powerful tool by which you can not only evaluate what could happen, such as the risk to develop a certain disease or the response to a particular therapy, but with metabolomics, you can especially assess causalities and the actual physiological outcome. But for now, we will focus on the microbiome, specifically the gut microbiome, that is gaining an enormous and still growing attention by researchers from all kinds of scientific disciplines. The microbiome has indeed dramatically reshaped our understanding of human biology over the last years, because the gut microbiome is increasingly recognized to function like an endocrine organ in the host. We will focus here on the bacteria living in our gut, as other microorganisms like fungi or archaea play rather a minor role. Here are some key facts about the human gut flora. When the gut microbiome is fully established at about 2 years after birth, there are up to 1,000 different bacterial species in the digestive tract. In total, we harbor approximately as many bacterial cells in and on our body as our own cells, which makes up around 2 kilograms of bacterial cells just in our gut, or about 100 times more bacterial genes than our own genes. Gut bacteria produce large amounts of metabolites. These are basically either metabolites derived from food or hosts that are biochemically modified by the microbes, or metabolites that are synthesized de novo. And since it was estimated that about 10% of all metabolites found in blood are in fact derived from the gut microbiome, we can say that blood is an excellent sample matrix to study the microbiome. In a symbiotic relationship between our microbiome and us, the gut bacteria exert many important physiological roles to maintain our health. First of all, they help us digest our food by extracting nutrients that we need to survive and for generating energy that would have been otherwise excreted. They synthesize vitamins such as B and K vitamins that are essential to us. They stimulate and train our immune system to recognize harmful invaders, and they can even outcompete pathogens, for example, by producing anti-inflammatory compounds. Already today, dysregulation of the gut flora has been correlated with a wide variety of diseases, including diabetes and obesity, neurological diseases such as Alzheimer's disease, cancer, and cardiovascular diseases such as atherosclerosis. In fact, there's hardly any disease that has not yet been linked to alterations in the gut microbiome. But what is needed are more functional studies providing causal relations. This is emphasized by an expert from the field, Professor Kani from the University of Louvain in Belgium, since there are, as he said, only a few exceptional studies that have investigated the role of microbial metabolic activities. Most studies conducted today on the gut microbiome traditionally apply 16S rRNA gene sequencing, but this has certain limitations. Usually, only fecal samples are analyzed, but not all bacterial species in the gut are also present in feces. Secondly, this technique captures only the bacterial composition, but there are also other microbes present in the gut. And most importantly, quantitative functional information is missing, since the absence of a microbial species does not automatically mean that all its functions are missing as well. Usually, there are other species that can take over and exert these functions. So it's more important to know which functions are present than which bacterial species are there. And this is where metabolomics comes into play. Metabolites derived from the gut microbiome can be quantified in feces, but also in other, more systemic matrices such as blood or urine, where there are no microbes present. Metabolomics captures the overall microbial metabolic activity, not only that of bacteria, and provides a complementary functional readout of microbial metabolism as well as its interaction with host and environmental factors. From now on, we should aim at combining both metagenomics and metabolomics to decode the full picture of a phenotype by studying functional microbiomics. As just mentioned, fecal samples are very limited in reflecting the situation in the gut. This was also shown in this Cell paper published a few years ago. The composition of the microbiome with regard to bacterial species is highly diverse along the different regions of the digestive tract that can reach a length of up to 9 meters. However, this diversity can only partially be reflected by stool samples, as depicted in the upper figure. And the same is true for the microbial functions inferred from metagenomics data seen in the lower figure. Even the most distal samples from the colon and rectum were significantly distinct in composition and function from stool. But maybe surprisingly to some of you, blood is in fact a very well-suited matrix to study the gut microbiome. A recent study could show that 77 standard laboratory tests and more than 250 plasma proteins fail to accurately predict alpha or Shannon diversity, that is a measure for the number of species and the number of gut bacterial cells per species. And the higher the diversity, the healthier your gut and your whole body. But a panel of only 11 plasma metabolites was indeed able to significantly distinguish individuals with low diversity from individuals with normal diversity. There are several more key advantages of blood that I'd like to mention here. Blood is a much more homogeneous sample matrix than stool, which makes also the sample collection much easier. And the availability of blood samples is usually higher, since they are anyway frequently collected for routine analysis. Also, the sample processing is easier and more standardized than for fecal samples. And metabolites that remain in the circulation may generally have an enhanced biological relevance compared to those excreted in feces. So now we know that many of the metabolites produced by the gut microbiome are shed to the bloodstream. But what is their role? In fact, the gut bacteria use these metabolites also for the communication with distant organs. The main communication channels are the gut liver, the gut heart, the gut brain, and the gut immune axis. For the gut liver axis, branched-chain amino acids and secondary bile acids play a major role, for example. And elevated levels of branched-chain amino acids can lead to obesity, insulin resistance, and type 2 diabetes. The gut heart axis is mediated, for instance, by metabolites of the choline pathway like TMAO and betaine. If the balance is shifted away from betaine towards TMAO, this is associated with an increased risk for cardiovascular disease. The gut-brain axis is facilitated, for example, by gamma-aminobutyric acid and tryptophan metabolites like serotonin. And the gut immune axis is mediated, for instance, by indole derivatives and short-chain fatty acids. This axis plays a crucial role, for example, if a cancer patient responds to an immunotherapy or not. So let's first have a closer look at the gut-liver axis and bile acid metabolism. Primary bile acids are produced in the liver from cholesterol and shared to the small intestine, where they facilitate the digestion of dietary lipids. But they exert also hormone-like functions by binding to nuclear receptors, such as the FXR, PXR, or the vitamin D receptor. This enables bile acids to regulate important processes such as glucose metabolism, innate immunity, as well as cell growth and differentiation. The majority of the bile acid secreted to the gut is taken up again, stored as bile in the gallbladder, and secreted again. This process is called enterohepatic circulation and takes place several times a day. In the gut, primary bile acids are converted by gut bacteria into secondary bile acids, which may also act as hormone-like signaling molecules, but mainly via G-protein coupled receptors such as the TGR5. But if the concentrations of secondary bile acids are too high, they can also exert cytotoxic effects. Since they are lipophilic molecules, they are able to intercalate into cell membranes and thereby destabilize the membrane and cause inflammation. A reduction of cytotoxic secondary bile acids in stool relative to primary bile acids was shown to be linked to non-alcoholic fatty liver and non-alcoholic steatohepatitis. The same was observed upon administration of antibiotics, which can also cause dysbiosis in the gut bacteria. If less cytotoxic secondary bile acids are secreted via stool, they might accumulate in the body where they can cause harm. In accordance with this, elevated levels of secondary bile acids compared to primary bile acids were also found in serum from Alzheimer patients, which perfectly fits to the cytotoxic effects secondary bile acids may exert upon accumulation. As you see, the ratio of primary to secondary bile acids is important, and also the matrix in which the bile acid levels are quantified makes a difference. So let's move on to the gut-brain axis and the tryptophan metabolism. Tryptophan is an essential aromatic amino acid and is mainly taken up via the nutrition, but it can also be produced to a certain extent from the gut microbiome. Tryptophan is not only a building block of proteins, but it is also metabolized into several bioactive molecules via three pathways: the serotonin, the kynurenine, and the indole pathway. Up to 10% of tryptophan is metabolized via the serotonin pathway. Serotonin is a neurotransmitter that is actually synthesized mainly by specialized cells in the gut epithelium. So knowing this, it is not very surprising that also gut microbes can modulate the availability of serotonin. Serotonin is one of the key molecules for signaling along the gut-brain axis, and in the central nervous system, serotonin exerts positive effects on the mood, sleep, and behavior, and it improves memory and also gut motility. Up to 95% of tryptophan enters the kynurenine pathway. This pathway gives rise to both neuroprotective and neurotoxic metabolites. But it is also an example for the gut immune axis, since this pathway can be highly induced by inflammation, and its activation leads to an anti-inflammatory response. Along the indole pathway, various gut bacteria metabolize tryptophan into indole and its derivatives. These metabolites are all signaling molecules of the gut immune axis. Some play a role in the immune homeostasis, and others maintain intestinal barrier functions. So overall, the gut-brain axis can play a crucial role in severe neuropsychiatric disorders such as depression or autism, as well as neurodegenerative diseases like Parkinson's or Alzheimer's disease. So how can we read out the interactions between the gut microbiome and the host? How can we study functional microbiomics? We now know that any changes in the gut microbiome can result in diseases including diabetes, neurological diseases, and cancer. These diseases are preceded and characterized by pathophysiological conditions such as inflammation, oxidative stress, or insulin resistance. These are in turn reflected by alterations in metabolic pathways such as the choline metabolism, the tryptophan metabolism, or the energy metabolism, which can all be read out by the MXP 1500 kit for targeted metabolomics. In total, the kit covers more than 600 metabolites and lipids and in addition to that, over 230 metabolite sums and ratios. The obtained data can then be used, for example, to predict patient stratification, treatment response, and potential side effects of an administered therapy. After this short overview on the human gut microbiome and its role in health and disease, we will now focus on neurodegenerative diseases, especially on Alzheimer's disease and the gut microbiome. Generally, Alzheimer's disease is a progressive neurodegenerative disorder and the most common cause of dementia in elderly people. It is characterized by the two main hallmarks of Alzheimer's disease: extracellular accumulation of the protein amyloid beta, known as plaques, and intracellular aggregation of the tau proteins into neurofibrillary tangles. And over time, this leads to a reduced brain volume due to a loss of synapses and neurons. Ultimately, this results in irreversible memory decline, change in behavior, and overall reduced quality of life. So today, Alzheimer's disease is diagnosed basically in two steps. First, questionnaires and cognitive as well as physical tests are conducted to assess a person's health status and medical history. In a second step, molecular description based on 3 parameters is recommended. These are amyloid beta, hyperphosphorylated tau, and total tau, together also known as the ATN classification. These markers can be assessed both in cerebrospinal fluid, CSF, and by neuroimaging. But only if all 3 ATN parameters are rated positive, that means above, or in the case of amyloid beta in CSF, below the respective cutoff value, and the clinical diagnosis from step 1 is positive as well, a patient will be diagnosed with Alzheimer's disease. Although the diagnosis of Alzheimer's disease based on CSF works quite well with an accuracy of 90%, there are certain disadvantages. The collection of a CSF sample is an invasive procedure. The diagnostic procedure is rather expensive, and it is suited only for advanced disease stages, not for the detection of preclinical or early stages. And that's why this approach is not very well suited as a tool for early detection or screening of Alzheimer's disease. Alternatively, the ATN markers can also be assessed by different neuroimaging techniques. But these do not always provide a clear diagnosis, especially in early stages of Alzheimer's disease. And since it is even more expensive than a diagnostic procedures based on CSF, it is also not well applicable for screening approaches. Focusing on the hallmarks of Alzheimer's disease has to date neither led to a really robust early diagnosis nor to successful therapy options. So maybe it is time for us to change our perspective. Can metabolomics probably contribute to brighten the dark? Can Alzheimer's disease be considered a metabolic disease? Could malnutrition of the brain lead to Alzheimer's disease? Is the microbiome involved in the pathogenesis? And does a change in perspective may lead to new treatment options? So these are the questions we'll try to answer in the remaining presentation. So a few years ago, the whole field was excited when this paper was published in Nature Medicine in 2014. The study was led by Professor Howard Federer from the Georgetown University Medical Center in Washington, D.C. And they have identified the first signature in blood that detects preclinical Alzheimer's disease. And this within 2 to 3 years before progression to mild cognitive impairment and Alzheimer's disease. And also with a very high accuracy of over 90%. But what is also interesting is that the signature seems to actually reflect pathophysiology. Cognitively normal persons that will progress to mild cognitive impairment and Alzheimer's disease had lower plasma levels of all 10 lipids in the panel as compared to individuals that remain cognitively normal. And this may be due to disruptions of the cell membrane integrity in neurons, which is an early feature of neurodegeneration that has already previously been described for Alzheimer's disease. Overall, since blood is easier to obtain than CSF and also less expensive than imaging, this metabolomic signature may be utilized as a screening marker for the detection of preclinical Alzheimer's disease. However, the results could unfortunately not be reproduced so far. But why is that? So Alzheimer's disease is a highly complex and continuous disease with different markers and symptoms arising at different time points during disease progression, as you can see here. So if you now want to validate a biomarker in an independent cohort, and that cohort has a slightly different distribution of the preclinical and clinical disease stages than the one that was used for the development of the biomarker, you will not be able to validate the marker with a satisfying accuracy. And this is actually also the reason why the field is still struggling with the verification of biomarkers at the moment. In order to solve this issue, the current disease stage of a patient would need to be defined more exactly first, to then design a respective cohort more carefully in which the marker is more likely to be reproduced. The importance of selecting disease stages more carefully is highlighted in this paper by Professor Rima Kadura-Daub from Duke University in the US, together with the whole Alzheimer's Disease Metabolomics Consortium. Based on samples from the Alzheimer's Disease Neuroimaging Initiative, or short ADNI cohort, they found that a metabolic profile in serum is specific to disease stage. In early stages of Alzheimer's disease, mainly phospholipids and sphingomyelins are altered, whereas in later stages, it is primarily amino acids and acid carnitines that are affected. And again, these alterations also reflect pathophysiological processes that are specific to the disease stage. The changes in sphingomyelins and phospholipids are related to alterations of the cell membrane structure in early stages, whereas the changes in amino acids and acid carnitines may reflect defects in the mitochondrial energy metabolism. These findings may help to define stages of Alzheimer's disease more exactly, which is required for the successful development of biomarkers and therapies for Alzheimer's disease. Based on the examples that we've just discussed, we can answer the first question, whether metabolomics can brighten the dark, with yes, stage-specific metabolic alterations may eventually enable early detection and prognosis of Alzheimer's disease. So does this mean that Alzheimer's is in fact a metabolic disease? To address this question, let's have a look at this paper based on the Baltimore Longitudinal Study of Aging, in short BLSA. It was published in 2018 by a team around Dr. Tambisatti from the National Institutes of Health in the US. Here we look at brain tissue from Alzheimer patients, asymptomatic individuals, and cognitively normal controls. We now focus on 3 different brain regions: the inferior temporal gyrus, short ITG, and the middle frontal gyrus, or MFG, are both regions that are heavily affected by amyloid plaques and tau panels in the case of Alzheimer's. And the cerebellum, abbreviated here with CB, is used as a control region since it is usually free from plaques and tangles. So in the control regions, there were no differences observed as expected. But in the 2 plaque regions, higher glucose levels were detected in Alzheimer patients compared to controls. So next, they wanted to see whether these high glucose levels were due to a reduced glycolysis. To address this, they had a look at the glycolytic flux reflected by metabolite ratios. The 1 ratio is a proxy for the rate-limiting enzymes hexokinase and phosphofructokinase, and the other ratio indicates the activity of pyruvate kinase, which is another rate-limiting enzyme of the glycolysis. Both metabolite ratios are actually part of a software tool, the Metabo Indicator, that comes with every MXP Quant500 kit. In total, more than 230 sums and ratios are covered by the tool. So now, what they found was that both the hexokinase and the pyruvate kinase activities seem to go down in Alzheimer's patients as the ratios went down, but only in the 2 brain regions where plaques and tangles may occur. So this means that the intracellular glucose turnaround seems to be lower in these regions, although sugar levels are higher. But how can this go together? There are basically 2 explanations. Either the activity of the glycolytic enzymes is reduced in these regions, or the glucose can somehow not enter the cells. So here's the answer. It is clearly shown that GLUT3, which is the glucose transporter of neurons and works independent from insulin, is significantly lower expressed in Alzheimer patients compared to cognitively normal individuals. GLUT1 is an insulin-dependent glucose transporter found mainly in astrocytes, and its expression remained unaffected. So this study provides clear evidence that malnutrition of an Alzheimer's brain is caused by lack of intracellular supply with glucose. And in fact, this result has recently been replicated in a large-scale proteomic study published in Nature Medicine. Overall, these findings suggest that Alzheimer's disease may be caused by an undersupply of brain cells with glucose. And besides that, it is generally known that insulin resistance favors Alzheimer's disease. Together, this might allow the conclusion that Alzheimer's disease may be considered as type 3 diabetes. With this, we have on the one hand outlined a possible way to explain Alzheimer's disease as a metabolic disease, and on the other hand also illustrated that malnutrition of the brain is a probable and conclusive hypothesis for the development of Alzheimer's disease. So next, we'd like to know whether the gut microbiome also plays a role in the development of Alzheimer's disease. And indeed, a series of papers based on almost 1,500 individuals from the ADNI cohort again describe the link between the gut microbiome and Alzheimer's disease. Here, elevated levels of secondary bile acids compared to primary bile acids were found in serum from patients with late-stage mild cognitive impairment and Alzheimer's disease. This was shown based on several ratios of secondary to primary bile acids. And since secondary bile acids can also exert cytotoxic effects by intercalating into cell membranes that could lead to inflammation, as discussed in the beginning, increased levels of secondary bile acids due to gut dysbiosis could be involved in cognitive decline and the development of Alzheimer's disease. And what is important to mention here is that this association could not only be replicated by an independent cohort, the ROSMAP cohort, but it was additionally proven also in brain tissue. So together, these results emphasize a role for the gut microbiome and the gut-brain axis in cognitive decline and the progression towards Alzheimer's disease. And a better understanding of these relations may contribute to an earlier detection of Alzheimer's and the development of disease intervention strategies. So now we can say, yes, the gut microbiome seems to be involved in Alzheimer's disease, and that alterations in the bile acid profile might contribute to disease progression. But will a change in perspective also lead to new targets or treatment options? So here's a hypothesis how Alzheimer's disease may arise. An unhealthy diet like the Western-style diet most of us consume can, over many years, induce several changes. First, the lipids from the diet lead to an unfavorable composition of the cell membrane and low-grade inflammation. But malnutrition also induces changes in the gut microbiome that can lead, on the one hand, to an increase in secondary bile acids, and on the other hand, to elevated levels of branched-chain amino acids. The secondary bile acids can not only destabilize cell membranes in the brain, but also alter energy homeostasis via the interaction with the nuclear receptors. Since this may lead to reduced intracellular glucose levels, brain cells now switch from glycolysis to fatty acid oxidation in order to maintain energy supply. The branched-chain amino acids induce insulin resistance, which in turn causes the release of triglycerides from your fat depots, since sugar as a fuel is not an option anymore. But over time, the unusual constant overload of the mitochondria in neurons with fatty acids results in mitochondrial dysfunction, loss of neurons, and impaired cognitive function. So overall, this schematic picture summarizes the evidence from the literature that malnutrition may induce an undersupply of the brain, which represents one probable way to develop Alzheimer's disease. So the concept that we've just discussed for Alzheimer's disease can actually also be applied to other chronic diseases. The main driving forces for both chronic inflammatory conditions and intracellular malnutrition are an unhealthy diet, and the resulting dysbiosis in the gut microbiome. So overall, we hypothesize that insulin resistance caused by malnutrition may eventually result in diabetes, but depending on the individual context, this might not be the only clinical endpoint. If you'd like to read more on the background of the discussed hypothesis, I can recommend these publications here. Or just contact me for further literature on that, or if you'd like to challenge that outline concept. So to address the last question, yes, we think that based on all the evidence from the literature, understanding Alzheimer's as a metabolic disease will eventually lead to targets beyond amyloid beta and tau. And with this, I'd like to summarize what we've heard today. First, we discussed that metabolomics provides a functional readout of the phenotype, integrating information from both the genome and the exposome. We've seen that the gut microbiome acts like an endocrine organ in health and disease development. We've learned about the main gut organ axis by which the microbiome communicates with distant organs using metabolites. And I've introduced you to the MXP Quant500 kit as a tool for functional microbiomics. We then had a look at the role of the metabolome and the microbiome in neurodegenerative diseases. We have seen that Alzheimer's disease can be considered a metabolic disease. We have hypothesized that microbiome-induced malnutrition of the brain might induce Alzheimer's disease. And we've discussed that the change in perspective could lead to the development of new targets and treatment options. So in conclusion, the good news is that if you consider neurodegeneration as a metabolic disease, it can, in many cases, be prevented by adhering to a healthy lifestyle, which also satisfies your gut microbiome. You can find more information on how to study neurometabolomics on our applications webpage on neurology. Or if you are more interested in how to study the microbiome with metabolomics, please visit our applications webpage on the microbiome. Thank you very much for your attention, and I'm happy to take your questions.